Angela is setting up her slides. I will introduce you to Angela Belcher. She's the Gurmishausen Professor of Materials Science and Engineering and Biological Engineering. Professor Belcher is a materials chemist with expertise in biomaterials, biomolecular materials, organic-inorganic interfaces, and solid-state chemistry. Her work focuses on evolving organisms to build new materials and devices for clean energy, electronics, the environment, and for medicine. She received her BA in Creative Studies and her PhD in Chemistry, both from the University of California, Santa Barbara. She's the recipient of the DuPont Young Investigators Award and the Presidential Early Career Award in Science and Engineering. She joined the faculty of MIT in 2002 after serving at the University of Texas, Austin as an associate professor in the Department of Chemistry and Biochemistry. She won the 2004 MacArthur Fellowship Award and the 4-Star General Recognition Award. The focus of her research is understanding and using the process by which nature makes materials in order to design novel hybrid organic, inorganic, electronic, and magnetic materials on new length scales. She uses these materials in a variety of applications: solar cells, batteries, medical diagnostics, and basic single molecule interactions related to disease. Her research, fitting to the Koch Institute, is highly interdisciplinary, bringing together the fields of inorganic chemistry, materials chemistry, biochemistry, molecular biology, and electrical engineering. She was named Scientific American's Research Leader of the Year in 2006 for her work using custom-evolved viruses to advance nanotechnology. And she was named a member of the Scientific American 50, the magazine's annual list recognizing outstanding technological leadership. In 2007, she received the Popular Mechanics Breakthrough Award and was honored by Time magazine as a climate change hero for her work developing a new kind of battery grown from tubular viruses. Please join me in welcoming Dr. Angela Belcher.
Thank you.
Thank you, Angela.
Well, I'm very excited to be here today to tell you a little bit about what we're thinking about in using nanomaterials to solve some of what we think are the biggest global challenges. Now, I didn't get that email that said keep it short and to the point, and so my plan is to try to link together the beginning of the Earth, batteries, and cancer in this time, that we have together. So my work is very collaborative. We work with people all over MIT. These are some of the collaborators of some of the projects I'll touch on today. And it's very exciting to be a part of the Koch Institute to really start pushing the boundaries of the problems that we are accustomed to solving. When I was first asked to be a part of it, I was intimidated, and I don't get intimidated that easily. But I thought, well, this is such an important problem, and there's so many great people working on it. You know, how can I possibly fit in? And then we kind of went through these cancer tutorials, you know, where I was learning about cancer biology, and I was thinking, wow, I need an easier problem to work on. But I'm going to hope to make the connections today with how we think about. I'm a material scientist. Everything to me is a material. And once I started thinking that cancer was a material, I started feeling a lot better in how to address these problems, and I'll talk a little bit about that today. So everyone has a periodic table that I handed out. I give these to the incoming freshman class every year. They say, welcome to MIT, now you're in your element. And so if you want to, I made them for freshman eyes, and so they get hard to read. On the other side are the amino acids and their pKa's, the pHs under which they change. And so I might refer to those as we go along.
Hey, Angie, did you give one of those to Barack Obama?
I do. There's a picture of Barack with it. And so in my group, we try to look at what are the most significant global issues, and is there any impact that we can have in these areas. And in my group, we're really interested in energy, healthcare, and the environment. And the intersection to us, why everything is a material to us, is we think of nanotechnology and biomaterials, that interface. How can controlling materials at the nanoscale affect energy, healthcare, and the environment? So that's the key that holds all this together. And everyone knows that nano is very, very small materials. And the kind of things that we focus on are solar cells, energy storage, batteries, CO2 sequestration and storage, imaging for medical imaging, therapeutics, and water purification. Those are kind of main topics that we're currently addressing. And so I call what we do genetic control of materials. Basically, how do you give genetic information to non-living structures so that non-living structures can enjoy some of the same things that biology does, so they can self-assemble, so they can correct themselves, so they can be in environments that are non-toxic. Those are the kind of things that we're interested in. And if you look at an abalone shell there, I did my PhD on how abalones grow shells, and they're beautiful structures, and there's all kinds of wonderful things about abalone shells, but they're made in the ocean. They're 98% by mass calcium carbonate, made out of chalk, basically. 2% by mass protein, but they're about 3,000 times tougher than their geological counterpart. But to me, the most exciting thing about an abalone is when a male and female abalone get together, they make millions of offspring, little baby abalone larvae, and they pass on to them genetic information that says, this is how to build an exquisite material. Here's the DNA. Here's how you make a material at room temperature, room pressure, that's non-toxic, and that's 3,000 times better than its geological counterpart. Same with diatoms, little marine organisms made out of glass. Every time they reproduce, they pass on the genetic information that says, here's how to make glass at room temperature at the nanoscale perfectly every time. And so what we say is, wow, could you do the same things for solar cells? Could you do the same thing for batteries? Or could you do the same thing for medical diagnostics? Can you make them smarter? Can you make them be able to be more lifelike? And so when we think about future technologies, here's the beginning of the Earth part. We think about the beginning of the Earth about 4.5 billion years ago. It only took about 1 billion years to have life. And this life rapidly became self-assembling. It had photosynthesis and did very, very complex things. But it wasn't until about 500 million years ago, during this geological time period called the Cambrian geological time period, the explosion of life, that organisms started making hard materials like shells and like glass. Why is that? They were already doing photosynthesis. They were already replicating and copying themselves. The reason is probably that they didn't have the opportunity. It was during this time there's increased calcium in the ocean, increased silicon, and these ions, these chemicals became toxic. And now the organisms had to learn how to deal with that. And by learning how to deal with that, they built really exquisite materials, diatoms and shells and bones. I always show this is my favorite biomaterial. This is my son standing next to his favorite biomaterial, the Triceratops. And anyone that has a 3-year-old know they're incredibly complex organisms. And so when we think about future kinds of technologies, we think about things simple, viruses and bacteria. Can we train viruses and bacteria to work with the whole periodic table so that we don't have just shells and glass? Can we have them make MRI contrast agents? Can we have them make batteries? Can we have them make solar cells? How do you give life to non-living structures? That's basically what we do. Oops. Now, going back to the idea of nano, you've probably heard that a lot of really interesting things happen at the nanoscale. When you take a bulk material, like a bulk material, and you start whittling it down to just a couple of atoms, the properties change. The magnetic properties change, as you just heard with Professor Seema's MR reagent. The optical properties change, the electronic properties change, a lot of things happen. So what we say is, what if you take things that are already really small, like DNA or an antibody or a small protein, and what if you have them be the manufacturer of nano devices or nano components? They're already really small and they're already very good. Let's have them work for us. What can a protein do for me? And so to us, the ocean is the reaction vessel that life came from. This is where life came from. This is where hard materials came from. We think life did very well. We think life could have done better if it had more opportunity. So you have iron and cobalt and magnesium and calcium in the environment that organisms could start building a toolkit out of. We say, let's give them the whole periodic table. Now, the thing I forgot to say is that organisms started making materials about 500 million years ago. It took them 50 million years to get good at it. And so I can't go to my funding agency and I said, I'm going to have a really good material, but I need 50 million years. And so we need a rapid way to think about it. How can you convince an organism or biology to start making materials on a much more rapid time scale? You know, even if I have 100 graduate students, which I don't have room for, it's still not going to work. Here's some pictures of natural biomaterials. This is a coccolithophoriid. It's a unicellular algae. This is an abalone shell. If you crack the abalone shell up, you can see that it's made out of these little tablets of chalk, basically. These are diatoms made out of silicon, SiO2, and these are magnetotactic bacteria that have small single-domain magnets. This is Fe3O4 used in navigation. This is how they figure out where they are in the water for getting nutrients and such. But the key is actually proteins. The key for controlling inorganic materials in our bones or in abalone shells or in these magnetotactic bacteria are the proteins. And if you flip your periodic table over, you have the amino acids. There's only 20 different amino acids that make up the proteins. And those amino acids differ from each other based on their chemical functional groups. Yet those chemical functional groups make it enormous, the pattern of them make it enormous or important in things like functioning enzymes in our body or the ability to grow material. So this is just a picture of a shell just showing that they're inorganic material, but they have this organic material, this protein in between that actually makes the difference between 3,000 times tougher or not. And the way this works in nature is that animals have, or organisms have proteins. These proteins have, in this case of a shell, really negative charged amino acids. So you turn your periodic table over, that's aspartic acid, glutamic acid. They grab calcium out of solution. They put down calcium carbonate, calcium carbonate. And we say, well, how can you take any inorganic material and force a protein to interact with it in a way that it can build an exquisite structure? And so we want to work with this part of the periodic table here, and this part of the periodic table. We want to work at all the periodic table, but these are the areas that we're focusing on right now. This is when President Obama came to visit recently. I got to meet with him for about 10 minutes, and I gave him a same periodic table that you are holding, and I've never seen anyone so happy to have a periodic table before. And he took it and he said, thank you. And I said, I just wanted you to have this in case you're ever stuck and you need to figure out a molecular weight. And you can pull it out of your pocket. And then President Hockfield's right there, and she pulls hers out. And she says, I have one too. And he said, thank you. I'll glance at it periodically. And so what my lab wants to do is take molecular specificity, things like antibodies that have evolved over a very long period of time, and maybe something like a semiconductor processor chip. How do you fit them together? How do you meld them together at that living, non-living interface? And what we do is we do a lot of experiments at a time. We do combinatorial approaches. We do 1 billion experiments at a time. And we decided to borrow the idea from the drug discovery industry and use something called bacteriophage. This is a virus. It's a virus that only infects bacteria. It's called a bacteria eater. It has a single-stranded DNA, and it's composed of 5 proteins. So by going into the single-stranded DNA and making a modification, basically cut, paste, put a new piece of DNA in, that DNA codes for a protein on some part of that virus. So a virus, this kind of virus is basically single-stranded DNA and protein. Now, you can go in and do that 1 billion times, and by doing it 1 billion times, you can make 1 billion different possibilities on this tip of the virus. So now, if you want to say, what kind of protein do I need to be able to bind to my iron oxide for my diagnostics, for my cancer diagnostics, we try 1 billion possibilities at once. We say, I think I have it in here. Hope I have it in here. We try 1 billion possibilities at once. You take about 1 billion possibilities in about 10 microliter solution. You force them to interact with any kind of material you want. So you're looking for that chemical specificity, chemical spatial specificity. Most of them get washed away, but one, really maybe 100 out of 1 billion will stick to that surface. Now you say, okay, I have 100 out of 1 billion. Viruses can't replicate themselves. They need a host. You have a host make copies of you. And now you have a population of viruses that have some affinity for this material through evolution it would have never interacted with before. You say, okay, now bind again. Most of them won't bind. You go through this process. And you're looking for 1 or 2 out of 1 billion that work. Okay? We almost always find 1 or 2 out of 1 billion that will do what you want it to be able to do. And so this is our toolkit. We love this virus because it's almost 1 micron in length and it's 6 nanometers across. It's nanometer scaled and micron scaled at the same time. And it's pretty simple DNA. You can go in and cut and paste changes in the DNA. You can make changes in proteins here, changes in 2,700 copies of the protein here, or changes in here. So it's a little toolkit. It's a toolkit that can do all kinds of things. And so I included this one because Tyler always mentions this. We actually used biology to look for something very, very rare event, which is atomic level dislocation in a semiconductor. So many people will know that the next generation solar cells, the next generation solar cells that we're going to have, hopefully, will be many different kinds of semiconductors that are stacked on top of each other. Okay, they're grown and stacked on top of each other. Now, when you take a material and you stack it on top of each other, it may not exactly match the material underneath it. And that leads to a defect. And those defects trap electrons and make things less active. So here's a defect. So if you grow these two materials on each other, there's a defect. My student said, I'm going to use biology to find an atomic level defect in a semiconductor. He's a really smart student. And I said, he said, I'm on full funding from NSF. And I said, OK, give it a try. And so he started with 1 billion sequences. And he said, bind this mistake, don't bind this perfect sequence. Bind this mistake, don't bind this perfect sequence. And he was able to find, he's all happy because he proved me wrong, 2 sequences out of 1 billion that could recognize atomic level dislocation in silicon. I thought, wow. For those people that are really interested in biology, thinking of it from a biological point of view, it's probably not that surprising because you think about the antibodies in your body that are working, trying to bind to specific targets or cells or proteins or something like that, and how specific they are. But you can actually get it to do something really different, like find a defect in a semiconductor. Or you can actually use it to try to find a defect in an airplane engine block. Okay? Because if you take an airplane engine block, it's a super alloy material. It's a material made of many different kinds of metals and metal oxides. And if you take it and you fatigue it, you take that metal and you bend it back and forth, it starts to have a little crack in it. Cracks are bad. But the chemistry at the crack is different than the chemistry of the surroundings. And through training a virus to bind or not bind, you can find viruses that only recognize cracks. And they can stick to your crack, and they can bring with it a fluorescent signal. And maybe one day they'll be able to bring with it the glue to repair that airplane engine block. So this is the kind of stuff that gave me a little bit of hope because we got really good at doing things like this. Well, maybe we can use this to do things like target cancer, which is a problem or a mistake, and then bring with it the ability to try to heal that cell or be able to solve whatever that problem is. But what we also think would be good is to be able to grow something. This is a virus that has a sequence that should be able to grow 4 different semiconductor nanoparticles on its head. You give it the semiconductor particle solution, and here's the virus here. It's just one little virus that grows a collection of semiconductors on its head. And this is about 1 million times magnified. This is actually what the atoms look like in that kind of structure. So we showed that you could actually grow something. But then what we wanted to do was actually grow whole wires. And if we actually went in and gave this virus not the ability to grow something on this end, but the whole length of the virus itself, now you could give it a material and it could grow a wire. And this is a beautiful wire because it's 1 micron in length and 6 or 20 nanometers in diameter. And so now we think about growing wires. And you do it by making multiple changes in DNA that code for the code of this protein. One thing I didn't tell you about viruses, another thing I love about this virus, is that it's crystalline, meaning that all the proteins in here are all crystallography related to each other. So if you can take advantage of that natural crystalline structure that biology gives you, now can use it to template something crystalline that's non-biological. Okay, you guys all still with me and seeing the connection? We're going from beginning of Earth to doing 1 billion sequences to look for something to solve a problem. And now we're going to solve one problem, which is a battery, and then we're going to look at cancer. You can go in and give it 2 genes to grow 2 different materials simultaneously. This is a gene to grow 2 different semiconductor materials. And so it's a new way of putting very different materials together. And so what we asked is, how do you give genetic information to a battery? How do you give a battery the sequence to become have higher energy density, to have higher power, to be made under non-toxic conditions and not explode? That's basically what we decided to go about and do. And it was kind of a tall order. In case you haven't had freshman chemistry recently, I'll show you how a battery works. This is a battery. You have 2 electrodes. This is a negative electrode called the anode. This is a positive electrode called the cathode. You guys are all carrying around lots of batteries right now. The batteries that you're carrying around on the negative electrode are made out of graphite or carbon. On the positive electrode, the batteries you're probably carrying around are lithium cobalt oxide. This has worked quite well for your cell phones and your laptops, but people will remember the exploding batteries, the Sony batteries that catch on fire. So we want to try to avoid that. So what we decided to use was do biology to engineer this part of the electrode and this part of the electrode, make it all at room temperature, use no toxic materials, and have it assemble itself. And so we engineered viruses to grow a cobalt oxide material. That's a material that's for a battery electrode. We engineered them to assemble themselves into a more complicated structure. And now once we get them, we're going to say, OK, can you become better? You did a pretty good job this time, but let's use evolution. Let's make them become better. What's your second generation battery look like? What's your third generation battery look like? What's your fourth generation battery look like? And so I'll just show you this. This is data. This is a virus that's grown cobalt oxide at room temperature. It's actually beautiful material, no toxic precursors at all. You grow them in a flask in the lab, spin them down, put in a cobalt source, and they grow these little wires for you. And you assemble them into a battery. Now, most of you guys are probably not used to looking at battery material, so I've referenced it to you to your current batteries right now. So this is actually how much energy you're going to be able to store. And this is your commercially available battery you're carrying in your laptop or your smartphone right now. And we thought we could go from 300, basically, almost to 1,000. So we thought we could get almost 3 times better. So we actually engineered our viruses to initially become 3 times better, but they start to fade over time. So as you go and you charge your phone and you charge, discharge, you want a rechargeable battery, right? Charge, discharge, charge, discharge, it's starting to fade. So my very smart student said, oh, that's because our virus only has 1 gene. What if we gave our virus 2 genes? What if we gave our virus a good gene? Remember in the diagram, lithium ions were going back and forth, right? That's part of the way that you're moving energy. But you have electrons going around the outside. And so my student said, we're good at moving lithium ions, we're not good at moving electrons. How do we move electrons better? And so they gave the virus a second gene, one to grow gold. You know gold's a good conductor. And so this is kind of fun. This is a transmission electron microscope image. This is a virus that has 2 genes. You give it gold, and it makes little gold nanoparticles. You see all the spaces in between? That's where it doesn't have the sequence to grow gold nanoparticles. That's where it has a sequence to grow cobalt. Now you give it cobalt, and it zips it up. And now it has a new structure. It's cobalt and gold. You test that as a battery electrode. 2 genes, 2 materials. First generation battery, second generation battery. Much, much better. It's actually better than what we thought was theoretically possible. But then that wasn't good enough because we wanted our batteries to self-assemble. So we started working with Professor Paula Hammond, a very great collaborator of mine who's also a member of the Koch Institute. And we said, OK, let's have a battery that self-assembles. So we took a glass slide, we dipped it into polymer. We had a virus like this polymer, and then we had it dipped it into a battery precursor material, cobalt and gold. And what we're able to do is pull out these batteries that are flexible and they're transparent. These are battery electrodes. They're transparent. My students don't give them to me anymore because I lose them all the time. But I think these are at both the MIT Museum and the Museum of Science right now in exhibits for these batteries. And they're made very, very easily. Let me tell you a little bit more of the story. I'm not sure how I'm doing for time. And this is a battery that recently went to the White House because it was such a good performing battery. And these are 2 very brilliant students of mine, both engineering students, who said, let's try to give viruses multiple genes to be even better. Let's have a virus grow now a positive electrode material and grab a single-walled carbon nanotube. Do people know about carbon nanotubes? Carbon nanotubes are, the kind of newer form of carbon. Nobel Prize for discovery of carbon nanotubes. The thing that's wonderful about carbon nanotubes is they're very, very skinny and they're very, very long. They have interesting mechanical properties, but they're really good at conducting electrons. But they're very, very small, and that's kind of a problem. But we engineered our viruses to go and pick up a carbon nanotube. Here's a virus, pick up a carbon nanotube and carry it, and now grow an inorganic material. Now we're going to ask our virus to wire itself back out so we can get our electrons out very easily. Okay? It sounds simple now. And then basically we're going to use to do something like light an LED. So here's what my students did. They got a virus that grabbed a carbon nanotube on its tip and then grew a good material on the outside and made a new conducting path for batteries. And basically with these carbon nanotubes on the tip of our virus act like a super highway to get electrons in and out very fast. And this is going to be important for high-power batteries. If you're going to use this for your car, you're going to have a high-power battery. You have to have a way of getting electrons in and out very, very quickly. My students figured out how to do that. They actually found 2 sequences out of 1 billion that could do this. Well, they really found 3 sequences out of 1 billion that can do this. And I was so excited about this, I felt like, I said, now I can retire. Because what they did was they found out that the power output of your battery was directly related to its DNA sequence. And that's what I've been living for so far, is having a DNA sequence that controls the electronic properties of your device. And that's what they did. They showed the tighter you bind this carbon nanotube, the higher power your battery is. It's very, very cool. And this is, in case you've never seen a virus binding the carbon nanotube, this is what it looks like at over 1 million times magnified. Basically, these little stripes here are carbon nanotubes, and our virus is all the way up here, and the virus is holding a little bundle of carbon nanotubes, and it's wiring it back to get the electrons out of your device. And this is a plot that actually is this amazing plot that actually shows that it's very high power. It was about an order of magnitude higher power than your batteries are right now that you have. It's very, and it's all made at room temperature. And you can make it in our undergraduate teaching labs. This battery went to the White House. It was very, very exciting. It was President Hockfield went to the White House, and President Hockfield and President Obama gave a press conference on clean energy technologies, and our battery was lighting the LED, and we weren't invited to the White House, my students and I, so we were watching it all on a laptop, and we didn't act, so it was kind of a funny story. My students, I got a call that says, we need one of your batteries to go to the White House. You need to put it FedEx today. And I had all this other stuff going on. And so I said to my students, we need a battery. And so they went and they built it really fast. They built this device, and they checked it a couple times. They didn't check it that well because we were up against a deadline. And so we're there, and the president walks out. Both presidents walk out. They flip on this, and we're all going, please, please work, you know. And then in the video, you can see our little beam of light that's lighting up the whole time. So we're very, very excited. And then, so we thought life couldn't get much better. And then President Obama came to visit us in the lab. And this is Paula Hammond, who she and I work on solar cells together. And President Obama came and wanted to learn more about our batteries and our solar cells. He was here to give a talk about green energy. And we spent about 10 minutes, him holding our devices and such. And it was really, now I'm saying, what's next? You know, it was really a really good time. I don't know how much time I have left.
You have about 15 minutes
awesome.
Talk about your topic, and we'll have some time for questions too.
Okay, so now how do we take what we've learned to work on a much harder problem, which is cancer? I mean, cancer is to me, I was very intimidated because, you know, I went to these talks, and Tyler and some other people gave a talk about, here's a cell in cancer, and here's all the different ways it can go wrong. And I'm thinking, wow, you know, I'm used to thinking about electrodes, and there's just not as many ways you can go wrong in building a solar cell or building a battery. And so we made 2 attempts in the last year on trying to say, how can we bring what we're good at towards trying to address some problems and issues in cancer. And the first part I'm going to talk about genetically templated magnetic nanowires for amplified targeted imaging. And again, what we think about, what we're really good at is giving genetic information to non-living structures. And so let's make diagnostic tools that have DNA in them, that are smart and can have multiple functions, and that we can change their DNA over time to give them new kinds of functions. That's basically what we're going to do. And the way that we started on that was with bacteriophage, but motivation is, I probably don't really need to talk about. We're really interested in metastasis, and about 90% of the deaths occur due to this. We're going to use nanoparticle probes for, in this case, magnetic imaging. And one of the problems with when you try to put small magnetic particles into animals or into people is that it's cleared very quickly. And so we thought, wow, well, we have this genetic tool, this M13 bacteriophage, and it's long and skinny. So maybe it will be able to be active longer within a structure. And there's also there are antibodies that are known as biomarkers, but it's difficult to identify early stage markers. And so our technique will give us opportunity to come up with new kinds of markers. And so what we did was we started with a prostate cancer cell line, and we used a sequence called SPARC, which targets a protein that's expressed on cancer cells. It's highly expressed in metastasis to the bone. But we took one of those sequences and we displayed it into the end of our M13 bacteriophage, the sequence that would recognize the SPARC sequence. Then we gave our virus the sequence to grow magnetic nanoparticles for MR imaging, and then we gave it the sequence to carry with it a therapeutic agent. So it's all encoded at the DNA. Here's some wires of our viruses that are binding these magnetic nanoparticles. These are iron oxide particles, and looking at the structure of these. And these are some of our first targeting in cells, not in animals at this time. And so what we did is, this is our negative control here, where they don't have a sequence to bind to the cells. And these are some prostate cancer cells down here, where our virus has the sequence to bind to that prostate cell, pass into the cell, and bring with it an MRI contrast agent. This time we're just looking at the light microscope, so it has the MRI contrast agent plus a fluorescent signal. And you can see that this green fluorescence is taken up by these cells. And we found out with these targeted uptake, we get about 50% improvement in uptake of these magnetic materials. And this is actually looking at nanoparticles. So now, as Professor Seema mentioned before, we're looking at relaxation time. And so we're looking at dark contrast agents. So we're looking at signal going down as being a positive result. And so these are nanoparticles by themselves. These are nanoparticles with this sequence. And these are our viruses with this sequence. And so they're actually targeted very well towards these prostate cancer cells that are taken up. And this is actually showing I like data. This is actually showing two different cell lines. This is a cell line that has a little bit of the prostate cancer target on it, and this is a cell that has a lot. And you can actually see that it becomes lower, so actually more is taken up, and it'll be easier to image. Now this is exciting for us because this data is from last week. This data comes from our first in vivo model of a mouse. Taking a mouse that has prostate cancer tumor, injecting it with our viruses that have multiple genes to do multiple things, target it towards that prostate cancer cell, and bring with it, in this case, just an MRI contrast agent. So I'm not actually really used to looking at these. This is my first in vivo experiment ever. And this is actually a mouse, and this, I think, is the spine of the mouse. This is actually a rather large tumor of a prostate cancer inside a mouse. And this is actually what it looks like. It's this bright structure. It's bright because it has a lot of fluid in it. And then this is the movie. Like I said, I saw this my first time recently myself. It's a movie kind of going through cross section of it, and you can see this basically tumor in the mouse. Now this is actually after adding our targeted nanowires. The things are going to get darker because it's decreasing the contrast. That's the kind of contrast agent this is. And this is the tumor afterwards, and it's much less bright. So it's actually showing us that we are indeed targeting this specifically, and we would be able to tell this on MRI that they indeed had prostate cancer. Then we went on to do other things, like now put a chemotherapeutic agent on this end, deliver it to a cell, be able to image it, and then watch that more cells were dying in the presence of this chemotherapeutic agent. This is very preliminary work. That's why my students said sneak peek. It's very preliminary. And so, that's kind of how we got in it, because we were good at making materials. That's what we do. But we had these circulating tumor cells meetings, and I was sitting there again in the back, kind of feeling sorry for myself because I don't know how I fit in. And Professor Hines was giving a talk about how circulating tumor cells bind to platelets in the blood. And I literally almost fell out of my chair because I got so excited because he showed this picture. I don't have his picture. He showed this beautiful picture of a circulating tumor cell that was surrounded by all these platelets all around the outside. And I said, wow, that looks like a materials problem, because that looks like a materials problem, because to me it looked like a unicellular algae that grows calcium carbonate on the outside to protect itself. And I said, aha. So what's actually happening here, and I mean, this is new to me, but the circulating tumor cells are protecting themselves with these platelets. And I said, let's think about how to exploit those platelets as a marker for early metastases. And so there's it's a very hot field right now, and people are looking at taking, this would be a circulating tumor cell here, and labeling with antibodies against a kind of marker called EpCAM, that's an epithelial cell marker, and trying to trap it onto a disc, basically, and count the number of very rare events, which are circulating tumor cell. And so what Professor Hines showed was that tumor cells are associated with these little platelet cells. And so now we have a big program where we're saying, okay, let's treat these platelets themselves as a way of diagnosing circulating tumor cells. and a way of also trying to, once you pull them out, to try to find new markers to be able to look for circulating tumor cells. And some initial data is really, really positive, and we did this in collaboration with Professor Jack's lab on using some of their known cell lines, where we can now pull out circulating tumor cells out of a spike solution of blood in a mouse. So we think it's very, very exciting. Forgot I had one more story. So just like we were able to engineer viruses to be able to grab on carbon nanotubes, which I said before, for batteries, we've started doing this, engineering viruses to grab carbon nanotubes for fluorescent imaging for cancer. And this is just a really cool picture. This is one virus that can bind about 20 carbon nanotubes around its tip. Again, this is a very high resolution image. This is 2 nanometers across. Okay, very, very small. And no one's ever done anything like this. It's pretty neat. But what we're doing is, normally when you take these semiconducting carbon nanotubes, they're fluorescent. They're really, really nice. But what happens is, in solution, they actually clump together. And when they clump together, they quench the fluorescence and they go dark. And normally how people deal with that is they put soap around them. You take a carbon nanotube, put a lot of soap around it. It's very fluorescent. It's very, very nice. We said, what if instead of using something like soap, which you can't use in the body, what if you use a virus to wrap up these carbon nanotubes and keep them all independent from each other, and now they're still fluorescent? And that's what we're able to do. And so I'm just going to skip ahead and spare you all the details of that, but actually just show you these fluorescent emission versus excitation of our carbon nanotubes are fluorescent after at least 72 hours, much, much longer in solution. So now we can take them and target them directly to cancer cells and do fluorescence imaging in the near IR. And that, I'm going to leave it at that and see if you have any questions or comments. Thank you.
You spoke about how the environment had all these toxic substances, and they found a way to deal with it or make something very special out of it. Do you see that happening again, that there are opportunities to take some toxic substances that have built out over time and turn those into positive elements?
Well, it's definitely interesting. So in nature, biology is really good at removing toxic things like cadmium from the environment and in making inorganic nanoparticles and sequestering them. In that case, it's not necessarily for the good, except for it allows them to be able to live. So I think that's definitely a possibility. That's a really good question. It's a very broad question. So I'm kind of flipping through my brain to think of other examples of that. But the only problem is that it takes such a long time. It happens on such a long time scale. But I think that this is an example. People say carbon nanotubes are great for so many things: structural things, electronic things, computing, fuel cells. But people are worried about them in the environment. We think that you can take a virus, engineer it to bind a carbon nanotube, and it makes it less toxic because it can't do the bad things that people think that they do to cells, poke holes in them and make leaks and things like that. So in this case, this is a directed selection evolution to that problem. But I'm going to think about that more because it's a great question, and that's my one example that I have. Yes?
By enhancing the luminescence in the near IR, I think of a number of applications, such as very much better X-rays or imaging of breast cancer and so on, enabling you to detect breast cancer much earlier. Is that?
That's exactly. So I'm showing you this data that's like 3 weeks old. So this is exactly where we're going with it. And I think my students put it together on the next slide. Kind of advantages of the system. Oh, there it is. So single-wall carbon nanotubes are really stable over time to fluorescence. They don't fade. They don't quench like dye molecules are. But what happens to them is if you have a conducting and a semiconductor nanotube, they come together in the body or something like that, and they basically cancel each other out. So what we're doing is we're trying to think of this as a way of replacing dyes for imaging. And we, the same experiments that we did with the prostate cancer cell line for MRI imaging, we're working on right now for fluorescent imaging. But we just submitted a proposal for doing a similar thing with breast cancer because we agree that we think that's a great place to be able to go. We haven't tested these out in cells yet. We haven't tested these out for the fluorescence in vivo yet. Yes?
Angie, it's fascinating to hear you talk every time you random thing. One of the things about tumors, which is quite exciting and interesting, is they actually self-assemble themselves, right? So we are full of cancer cells all the time, and our body does a fairly good job at getting rid of them. And it's pretty well understood that at some point these guys sort of bunch up together and organize themselves and say, okay, let's start building the structures. And that's what actually causes a tumor, and they build vascularization and whatnot. Would it be possible to try to figure out what were the genes that triggered those self-assembly, try to replicate those said another way, in order to identify which genes allow them to self-assemble, would it be possible to use your viral system in some way that would give you hints as to which genes should be targeted to prevent cancer cells to organizing and self-assembling?
I think that's a great idea. I think that that you could kind of think of the viruses maybe as knockouts, and knockouts to maybe self-assembly. And so I definitely like to think about that more. What I would have trouble with is figuring out the genes that led to cancer cells self-assembly. I have no idea how to do that myself. But I could think about ways of, if we have candidates, using this system and using material systems to try to probe that and pull it apart.
Because the candidates exist, right? I mean, there's an exhaustive list of all the genes that could be the oncogenes. So the challenge is, can you test them and figure out which are the ones that originate the self-assembly? And then if you can target those, then you can sort of start thinking about preventing cancer.
I mean, I would love to do something like that. And like I said, I'm on a steep learning curve, and I'd love to learn more about it. It sounds interesting. We just have a new robot in our lab to be able to do 1,000 screens so take a billion possibilities, narrow them down to 1,000, and then ways of combinatorially screening those 1,000 for functions. And we're trying to figure out how to fit that into looking at cells and things for circulating tumor cells. So maybe maybe our new robot is a way to do it.
Can I make a comment and a question? The comment is, for me, recruited Angie into the CodeKit, so you can see why I was interested in recruiting her. Coming from material science, being such a creative person, and how satisfying it is for me to see the first in vivo experiment, which isn't after too long, actually, that you're delivering on the promise that I was banking on there. But I would imagine that another advantage of using phage evolution is developing phage that have dual specificity with respect to different markers. Because one of the challenges in cancer is differentiation and specificity of the imaging agent to tell this one from that one.
Like barcoding.
Yeah, exactly. So have you gone in that direction at all in these?
Well, we tried a little bit in the circulating tumor cell project, and I can just tell you that cancer cells are a lot more difficult than single crystal semiconductors. That's one of the reasons that so the problem is we get lots of hits. And then how do you check those hits for how good they are? And for us, that was a challenge. And now with our new robot, we're hoping to be able to test 1,000 hits and kind of differentiate them in an easier way. And that's exactly where we're going, because it's that power of positive and negative selection to be able to get at kind of a yes or no event. And that's exactly where but your group's working with us a lot on trying to help ask those questions, right?
Another question relates to sort of the practical application. People have used the evolved phage protein in the body, but I don't know if people have used the evolved phage in the body. Has that been done, and do you know if there are regulatory limitations?
Well, I mean, it's going to happen soon, because I'm watching this kind of pop up. Now, people have done have used phage libraries for selection in cancer patients, late-stage cancer patients, and have used that as a way of screening, and then I guess you take a biopsy and pull it off and find out clones that way. Now, phage are becoming in, actually, and they're being used for other kinds of therapeutics, for antibacterial, and they're used in cholera somehow. I'm not exactly sure how that is, but now people have shown that in vivo selection in people, you can pull out positive targets. Now, the thing that's interesting is they've also shown that they're non-toxic, but they are immunogenic.
I've seen a group developing some phage to go to the CNS to block formation for in vivo applications that are on there.
Yeah. Yes.
Is your fighting Durkin phage as immunogenic by itself?
It is. It is. So this study that I was just stating is that if you give it back to cancer patients, they will have antibodies to it. But it was immunogenic, but not toxic. But remember that with a lot of the structures that we're targeting to mice are completely covered in an inorganic material. In that case, iron oxide, and so it might be sticky. But we haven't looked at. That's a great question. We haven't looked at that in terms of response to the iron oxide, because it's not going to really see the phage, because the phage is completely coated. It's going to see the inorganic material. That's a really good question. So I thank you for your patience and your attention, and I'm happy to talk more at the reception tonight. Thanks. If you didn't get a periodic table, there's more up here. Or if you want to take one to a friend.